Hello, thank you for coming. My name is Anatoly. It's a great pleasure to have a talk about something inspirational, intriguing, and promising, about a chance to live more, probably much more. There is no proof tool to do it, however, in my opinion, it could be a bacteria in permafrost. Well, life is beautiful, isn't it? However, death is inevitable. Most long-living creatures are better than humans, but still it looks like the maximum is about 200–300 years or little more. When I was young, I asked myself, is it something what we can do about it? Humans, as many other species, consist of billions of cells. Some born, some die. Generally, we die because cells are dying. So we start with a simple question. How old can be a single cell? If it lives longer, we could live longer. All cells are dying. Even bacteria are able to age. Still, how long can be a life of a single cell? As a geologist, I know there are deposits which are formed long time ago. For example, salt deposits. They were formed about 300 million years ago, and water or air could not go inside of them because they are solid and come to solution if water contact. So it's difficult to contaminate them. Everything what is inside came to us from the time of millions of years ago. And those deposits are widely distributed, so we have a nice opportunity to sample them. It was a paper published about 20 years ago about finding a live bacteria in that permanent salt aged 250 million years. Surprisingly, genome of that bacteria was similar to the modern bacteria. That stays unexplained because there is so-called molecular clock, which I'll discuss a little later. Microorganisms evolve with time, so ancient genome and modern genome of the same bacteria must differ from each other. Now it's a hot discussion. Many scientists reported that finding is wrong. It is contamination or wrong age estimation. It is hard to believe there is a living creature having the same biochemistry as we are, but which is able to live millions of years. Impossible. Since that first publication up to now, you can find the tens of similar papers. And a number of similar findings of bacteria in ancient salts. China, Europe, labs in US, and so on. So it's not a myth anymore. Bacteria remains viable in salts for millions of years. Well, it's really difficult to find them. That is why some skeptics are still there. But it is unlikely that quite a few labs are making mistakes. Here how these ancient bacteria in salts look like. They come to us from deposits aged hundreds of million years ago and still alive and feel good. It is hard to believe they were acting during all that time. Did they sleep somehow, or kind of real rest state, or what? It is unclear. Ancient salt, it's not the only environment where the bacteria with ability to long life exists. Another example, amber, with age 25, 40, even 100 million years old. Bacteria trapped in ancient amber were coming back to life also. Still there are skeptics about the possibility of life lasting for millions of years. Another discovery has been made in Antarctic ice by Russians more than 30 years ago. Different microbial species were found there alive at the depths of more than 3 kilometers beneath the ice shelf, and its age was evaluated up to 500,000 years. And, however, the temperature of ice is low, below minus 10 degrees Celsius or less, so no wonder the bacteria survived. It is known if you freeze the bacteria, it can be kept for a long time. At low temperature, even some human cells can be kept for a while. It is an interesting book called Geological Microbiology. It has been published in Soviet Union more than 60 years ago. The book states the bacteria is everywhere, deep underground, in a small amount, just a few cells per cubic centimeters, but in many types of sedimentary deposits. Here is a modern example. Microorganisms in Golden Mine, 2.8 kilometers deep in South Africa. Well, these microorganisms are not necessarily ancient. The cells could be possibly brought by the underground waters recently. And this is example is resident microbes in a 3 kilometers deep copper and zinc mine in Ontario, Canada. There is no proof the microbes are really old, though. In the same time, we have an excellent environment to look for ancient microbes, permafrost. Why? A few reasons. First, it is a huge territory. It's about 25% of the northern hemisphere occupied by permafrost, and it is up to 1.5 kilometers deep. Second, deposits were accumulated and kept at relatively low temperatures, so there is a lot of organic material which is only decomposed in part. The third, age of permafrost, or time of staying permanently frozen without thawing, is tens and hundreds of thousands and even millions of years in some areas. And of course, the number of microorganisms is surprisingly high there, so we can easily repeat sampling and confirm the presence of microbes, what is always difficult to do in case of ancient salts or amber. Here is exposure of coastal cliff at Barrow, Alaska, most northern part of North America. You can see ice wedges formed at the time of marine deposits accumulation. Structure is permafrost is stable. If permafrost melts, it changes the structure. For example, the ice wedge on a picture will lose its shape. So we're sure about the ground staying permanently frozen. We can evaluate its age studying historical geology, paleogeography, isotopes, etc. Water or even air, as well as microbes, cannot penetrate into the permafrost. Its permeability is extremely low. Therefore, any bacterial cells trapped in permafrost have at least the same age as the permafrost itself. What is also important, permafrost temperatures are not low enough. As you can see here in a picture presenting freezing of bacterial culture, in this particular case it freezes at the temperature lower than minus 20 degrees Celsius. We need at least minus 8 degrees Celsius or so to freeze the bacterial cells. The temperature of permafrost are normally in a range between minus 3 and minus 7 something degrees Celsius or often higher. So it's not low enough to freeze the cells. Bacteria in permafrost are not frozen, contain all chemicals in solution as usual, continuing its metabolism, just probably a bit slower. Sometimes permafrost is almost a pure ice, as that picture taken in Siberia. For bacteria, there is no way in as well as no way out. It is trapped in ice, and it is not frozen. like a time capsule brought to us from the past. We have to be careful sampling though. We cannot bring modern microbes in a sample during the sampling procedure. We cut the large cubes of frozen soil or ice, then have to put them in ethanol solution to clean the sample surface, and after that even take the samples into flames for a few seconds to make sure nothing alive left on the sample surface. We study then what is inside. On electron microscope pictures taken from permafrost, we see rod-shaped bacteria, red arrow shows where is it, attached to a mineral particle and partly trapped in ice. It does not look like the bacteria is able to move there, as well as grow or multiply at these conditions. Sometimes bacteria is just trapped in an ice crystal. Thin lines, borders of ice crystals, and one arrow shows the ice crystal border, another arrow shows where bacteria is. Bacteria has been identified using spectral analysis. Yellow peak of carbon shows that. Bacterial body has been found also covered by substance. Content of this cover includes high amounts of calcium and chloride ions. It means that bacteria collects cryoprotectors. This salt solution does not freeze at low temperatures and creates unfrozen water film around its body. That cover around bacteria trapped in permafrost probably contains also polysaccharides. That cover certainly plays a protective role. We also sampled an ice wedge in Fox permafrost tunnel in Alaska. The age of the ice have been estimated by radiocarbon about 25,000 years. It is relatively young permafrost. Samples were surface sterilized, melted and spread on agar media, and DNA was extracted from the melted sample. Plates were incubated for 3 months and about 300 different bacterial and 18 fungal colonies were received. Phylogenetic trees were constructed. There were different classes of bacteria, including new species. The closest relative of many representative strains originated in cold or frozen environments. These results suggested that a quite limited phylogenetic group of microorganisms were survived in the ice wave. The sensitivity of isolates to sub-zero temperatures was also examined. We found that some grew at -5 degrees Celsius. These Acrobacter bacteria were growing particularly fast. However, do they grow inside of the permafrost? It is unclear. Let's discuss that important question a little later. Up to now, a number of cases of bacteria found in permafrost and ice were reported. Here are some locations. Looking at the map there, no doubt the bacteria in permafrost are widely distributed. So we have plenty of opportunities to sample them, study and confirm the origin and age. It is interesting that even viruses have been found in permafrost. In the young permafrost only though, and mostly bacteriophages, viruses infecting bacteria. Now let's talk about the ability of this permafrost bacteria to grow inside of the permafrost. Bacteria need water for growth, constant supply of nutrients, as well as metabolites to be taken out. Is it possible during thousands and millions of years for cells trapped in permafrost, unable to move? Where is that stock of nutrients or area of garbage collection? Another story is the bacteria culture dynamics. They grow fast at the beginning, then slowing down and eventually die due to metabolites poisoning and bacterial aging. So how, without the space, nutrient stock being trapped and with a tendency to die, do these permafrost bacteria survive for a long time? It has been found that bacteria is able to slow down metabolism significantly. We don't know much about that kind of life, but they are still alive. If bacteria grow inside of permafrost, and what we see are not original cells trapped in frozen ground and ice years ago, but their descendants, in this case, we would see many cells close to each other, colonies, but we did not see colonies of bacteria in permafrost. We made hundreds of similar pictures, and everywhere just a single cells. So, if bacteria in permafrost are not frozen, does not grow, came to us from the past, which is thousands and millions of years ago, how can the bacterial cells be still alive? Why I am asking this? Because in DNA molecule, more than a few breaks a second occur. Because DNA is a major molecule keeping our data, and many other molecules inside of the cells are not stable enough to survive for a long time. DNA damage occurs at the rate minimum 10,000 nucleotides per cells per day. DNA is simply breaking too fast to survive for a long time. That is a major reason for aging of cells. And as soon as the cells are not frozen and subject to damage, all cells are aging and dying. There are many sites in DNA which are affected by chemical processes and damages. Free radicals, oxidation, hydration, even small level of radiation kills DNA. One of the most common way to damage is cytosine deamination. It interacts with water, producing uracil, which makes DNA not functional anymore. This process of hydration is inevitable, because there is always water in the cell. DNA is hydrophobic and structured, preventing water to come in. But it is hard to make that protection good enough, especially if time is long. Time of half-life of cytosine in water solution is just 500 years, even at low temperature, because thermal fluctuations of molecules destroy it, without any free radicals, radiation, etc. It means within 500 years, half amount of all cytosine in cells possible will be destroyed or decayed. Probably that is why a maximum lifespan of living creatures are close to that value. We have to take into account powerful repair mechanism working in the life cell though. These mechanisms repair majority of damages, but the mechanisms are not powerful enough to keep DNA stable, especially for a long time. These steady-state DNA damages are actually a cause of aging. Older cells have more DNA damages than younger cells. How fast DNA decays is still not very accurate calculated. One of the reasons because it depends on environmental conditions. However, it is tens of DNA sites per cell per year. It means for a million years, no single gene survived. Look at some calculations and data published. If we take just a sequence of 100 nucleotides or base pairs, no such a sequence will survive for a million years, even at temperature minus 10 Celsius. Surely not a gene which is about 1,000 base pairs. They were trials to amplify ancient DNA by PCR, polymerase chain reaction. The results show no PCR products for million-year-old samples because its DNA was completely destroyed. Therefore, genome as well as the cell survival is limited. That is why plants, animals, and humans die relatively soon, even having powerful repair mechanisms. So how bacteria in permafrost is still alive for thousands and millions of years? Let's have a look at the area of oldest known permafrost in the middle of Siberia, central Yakutia. It is a pretty cold place, which has been never covered by glaciers, so permafrost there was formed, by our estimation, probably about 3.5 million years ago. The place is called Mammoth Mountain due to many mammoths' remains found there. An exposure consisted of river sand and upper layers of ice ages was sampled. Sands were deposited in Niagen approximately 12 million years ago and became frozen later. Temperature changes in northern hemisphere were studied relatively well. Here are some results showing temperature, and vertical line is the modern temperature level. About 3.54 million years ago, temperature was about the same as now. Now ground temperatures in the area is -3, -5 degrees Celsius, and wide distribution of permafrost. That is why permafrost there is one of the oldest in the northern hemisphere. There is a high cliff here, and deposits together with sand contain clay layers with ice lenses. We found bacteria inside these deposits. They were trapped in permafrost as single cells. Sometimes we could see them under electron microscope inside of ice. The bacteria were able to grow. On the left you see them in petri dish, right under optical microscope. It is a positive gram-staining rod-shaped bacteria related to Bacillus genus. We did a full genome sequence of the bacteria. We named it a Bacillus F at the beginning. We found the Bacillus F has a high level of homology to Bacillus cereus, well-known organism, widely distributed. The difference in DNA sequences between these two strains is comparable to the difference between sequences of other Bacillus cereus strains. So it's actually a strain of Bacillus cereus. However, these two strains, Bacillus F and cereus, still have some differences, as you can see here from the plot of genome comparison. Most important, Bacillus cereus is pathogenic. Our Bacillus F is not pathogenic. Our Bacillus F does not have genes responsible for pathogenesis. There is a mystery here. Ancient Bacillus F is so close to modern Bacillus cereus. How it could be if mutations occur? With time, any organism is getting mutations. If Bacillus F is old, but the compared strain of Bacillus cereus is modern, the last one should have mutations. There is so-called molecular clock, changing DNA with time, like a globulin here. And the clock is believed to be accurate. Remember, we already saw the same when discussed bacteria from salts and amber, where the same phenomena was. According to molecular clock theory, DNA sequences show divergence rates of about 1% per million years in bacteria, mammals, and plants. However, some genomic regions were changing considerably slower, like 1% per 50 million years. Functionally important parts of amino acid and DNA sequence evolve much more slowly. Example you can see here in a table: gene for growth hormone and others. How does it happen? Bacteria have a very old origin. They came from the past and have passed their long evolution, 3.4 billion years and older. They had time to choose the best way of life. Bacteria are simple, and they use simple substances to consume. So they didn't have many ways to choose the best way of life. They were tried and chosen the best possible genes helping them to survive. You see what happened? It looks they have found the best genes for life a long time ago. And for the last billion years do not create any new genes, but just exchange them. How they keep their useful genes stable? Some people say recreate them. Probably it's easier to keep them though. It could be a mechanism to make stable genes and genome as a whole. So those who survived have that efficient mechanism of genes protection. If they keep DNA stable, then they have found a cure against aging, because the aging it's a gene damage. And if they found a way to protect their DNA, they can survive extreme environment. How about the space? Possible. Here are traces of bacteria-like forms in meteorites. Interesting. Some impact structures created as a result of meteorite impact contained a lot of microbes. If the bacteria have such a useful for survival mechanism, why don't we also use it? There are bad and good microbes. Some of them prolong life. Words of Ilya Mechnik fit our case. He tried to prolong life uh, also taking lactic bacteria. Nowadays, lactic bacteria probiotics are popular worldwide. What are the properties of the bacteria from permafrost? Can we use it for ourselves right away? Probably. Our bacteria, Bacillus F, is not harmful and not pathogenic. Our bacteria was tested on flies. Mortality of flies imago grown on media with bacillus is less after heat shock and after ultraviolet exposure in comparison with the control group. It means our bacteria protects against heat shock and radiation. It actually has a number of other useful properties. It is extremely resistant. It stimulates immune system and prolongs life. It shows antimicrobial activity against pathogenic strains like Salmonella and others. We observed a significant increase in physical strength in mice after injection of Bacillus F. And the most curious result, a number of men volunteers were taking Bacillus F as a probiotic food supplement, and we found increased levels of testosterone after taking in all cases. So what protects DNA of this bacteria against damage for a long time? What Bacillus F has, what we don't have? It is a very promising study, I think. As a conclusion, I am sure a mechanism of extreme survival of DNA genes exists. It could be two possible ways of survival DNA. First, conservation, like binding proteins, organic stable molecules, etc., and extremely efficient repair mechanism. Modern biotechniques may help to find proteins in the bacteria under cold stress and therefore to identify that unknown mechanism. The isolated bacteria has important useful properties. However, if we knew how the bacillus F protects DNA, we potentially could find a mechanism of protection of our own DNA. That is a way to eternal life. Why don't we go there? Thank you very much for your time, which is priceless. From other hand, who knows? Perhaps it helps to increase your lifetime.